All right. Thanks for the introduction. And hi, everyone. Thank you for coming. I'm Neil, and today I'm going to talking to you about, one chapter or one part of what I've been thinking about, but something that has sort of guided everything that I've thought about and that ended up in my PhD, and that's leveraging evolutionary information to improve protein modeling. And first, those of you who know me are going to be completely unsurprised. I'm going to do a little tangent about a short story called The Library of Babel. So The Library of Babel is a short story written by Argentinian author Jorge Luis Borges in 1941. And it's about a fictional library whose bookshelves contain all possible combinations of the 22 orthographic symbols, a number which, though unimaginably vast, is not infinite. That is, all that is able to be expressed in every language. It's larger than the universe, and it contains all possible books. And those who live in this library go mad trying to look for everything they could ever want. Their biography is there, as well as an infinite number, or almost infinite number of forgeries of their own biography. So in exploring the space of proteins, see, it's relevant, tangent is relevant. Exploring the space of proteins, we find ourselves browsing in a similar library. In fact, I stole this analogy to Borges from Frances Arnold's 2018 Nobel Prize acceptance speech, where she describes her techniques for laboratory evolution. But instead of books, our library is full of proteins. And instead of stringing together orthographic symbols, we string together amino acids, of which there are 20 genetically encoded amino acids, and each have different biochemical properties. Some are charged, some are positively charged, negatively charged, hydrophobic, hydrophilic, and these properties determine how they interact with their environment. And each amino acid can be represented with a one-letter code. So here, the peptide serine, histidine, cysteine is represented as SHC. Combinatorics is fickle, and the number of possible proteins of length 100, which is relatively short even for a protein, is greater than the number of atoms in the observable universe. Thanks, Mom. I'm going to start with the prologue, which is making all the colors of the rainbow. So we'll talk about what's a protein and why we want to engineer them. And our focus will be on proteins that fluoresce. So a protein is a polymer made up of molecular units, amino acids. They're bound together in a chain. This linear chain of amino acids folds in three-dimensional space. Locally, it twists to form helices, twists on itself to form sheets, and this we refer to as the secondary structure of the protein. Those local structures then further contort to form the protein's tertiary structure, which is the 3D coordinates of every atom in the protein. This tertiary structure, along with its dynamics, are what dictate the chemical functions that the protein can perform. So the sequence is informationally complete. And what that means is the sequence of amino acids contains all the information necessary for the cell to fold it into the state where it performs its function. And we can think of every naturally occurring protein as nature's solution to a biochemical problem. So how do I transport oxygen through the bloodstream? Hemoglobin. How do I glow green? Nature found a barrel structure with a fluorophore in the center. This is the green fluorescent protein. Sometimes nature gets feisty. It asks, how do I bind the ACE2 receptor and crash the stock market? It's the COVID spike protein. So I'm going to examine a short story of fluorescent proteins to show why engineering proteins is so valuable and examine some of the challenges that are involved. So let's take this green fluorescent protein. What if we want it to glow yellow, orange, red? How would we find the appropriate protein sequence that accomplishes this? And this isn't just a thought experiment. Fluorescent proteins have unlocked a vast array of applications, and I'll just name two. Fluorescence microscopy allows us to tag different parts of the cell with fluorescent proteins, the nucleus in blue, microtubules in green, and watch how cells develop and how they respond to disease. The more colors that we have, the more portions of the cell we can watch simultaneously. Fluorescence activated cell sorting allows us to tag a certain function with a fluorescent protein and sort cells that perform that function from cells that don't. It's a critical tool in protein engineering. So for every protein engineering campaign, we need a starting point. And here's where we look to evolution for help. So naturally occurring genes coding for fluorescent proteins exist in a wide array of organisms. The original GFP was discovered in 1992 in crystal jellies and was used as a blueprint for further development. And a major breakthrough was finding new fluorescent proteins in other species. So red fluorescent proteins were derived from red proteins in the species of disc anemone, as well as orange proteins found in a species of stony coral. And molecular biologists have taken this naturally occurring function and optimized it for their own uses, which is completely different from the function that nature originally selected it for. The fluorescent proteins that we use today are the results of decades of engineering work. This particular timeline only goes to 2006, but the work continues to this day. And this engineering work was done to optimize the properties of fluorescent proteins to make them easy to work with in a laboratory. We want them to be bright and easy to image, much brighter than would be practical for an organism. We want them to be stable, and we want them to be small so that they're easy to attach and don't require complicated assembly within the cell. To accomplish this for the original red fluorescent proteins, it took 33 mutations from the disc anemone protein, modifying 15% of the sequence. These mutations were iteratively discovered over the course of multiple campaigns over 5 years of research. Engineering proteins is hard, and it's an amazing feat of scientific progress. With decades of research, biologists were able to use their understanding of the structure of GFP and the way that it functions to propose mutations. Decades of guess and check through the process of laboratory evolution, screening mutations to see if they'll work. And each determined structure required a complicated process of purification and microscopy. And every newly discovered fluorescent protein was probably an exotalk like this one. And this is for one of the most well understood proteins of all time. So what happens if I want to explore a new function? What if I want to unlock new chemistry and biocatalysis on a rainbow of different substrates, develop new therapeutics to be delivered to different parts of the body, develop new molecular tools like molecular scissors? We don't have decades of research, detailed functional understanding, or high resolution structures for every single function that we may want to investigate. So what are we to do? How do we proceed in the face of limited data? So this is where we turn to machine learning with a model that can map from sequence to function to help us figure out how to create new designs. And the two questions that we want machine learning to answer, are, where do I start, which I'll call discovery, and how do I improve, which is I'll call optimization. And we want to use our models to guide our designs towards new diverse solutions and have fewer campaigns that fail due to dead ends. Okay, we've arrived at the end of the prologue, and I'm going to outline the remaining chapters of our story for today. So in chapter 1, we'll talk about learning the language of proteins by leveraging evolutionary information. In chapter 2, I'll argue that protein language models have come of age, focusing on the question of structure prediction. And in chapter 3, we'll conclude by outlining protein design as the next frontier for machine learning. So let's start with learning the language of proteins. How does nature find a solution to a new biochemical problem? How do we get hemoglobin, green fluorescent protein, COVID spike proteins? These are all the result of the natural process of evolution. An essential concept here is homology. So here I've shown homologous structures present in mammalian forelimbs. Each of the color-coded bones has been derived from a common tetrapod ancestor. So these are all variations on a theme, and homology gives us information. By looking at a human arm, we can learn something about the structure of a bat wing. And the reason all of these structures are related is that nature is a tinkerer, not an inventor. Genes mutate, accumulating changes, insertions, deletions, which are then selected in the arena of organismal fitness. And the secret to success in evolution is the same as the secret to success in academia.
In one word, he told me the secret of success in mathematics. Plagiarize. Plagiarize. Let no one else's work plagiarize. Remember why the good Lord majorize, so don't shade your eyes, but plagiarize, plagiarize, plagiarize. Only be sure always to call it, please,
evolution. So what does homology look like for proteins? We can set up a taxonomy for protein structures like we do for organismal kingdoms. So the structural classification of proteins, or SCOPE database, organizes structural similarity with the following taxonomy. Proteins in the same class exhibit the same secondary or similar secondary structure composition and order. Proteins in the same fold exhibit gross structural similarity. Proteins in the same superfamily are hypothesized to have a common ancestor. And proteins within the same family have a clear evolutionary relationship and high sequence similarity. So for example, the phosphate binding enzymes in this superfamily that are displayed all are derived from a common Tim barrel fold. So how do we use evolutionary relationships to make efficient use of labeled data? If you get to see a collection of all my second cousins, you're going to know a little bit about me. That's the intuition. So let's walk through where evolutionary information comes from. we'll take a cartoon loop protein with 4 cartoon amino acids, blue followed by red, then yellow. And the loop is pinched off by this interaction between blue and yellow residues. Let's say that this loop is somehow important for organismal function. Then during the natural process of DNA replication, modifications accumulate, mutations, insertions, deletions, but the essential function of the protein, in this case, its loop structure, is preserved. And we get to observe each of these organisms in the present day with their genomes deposited in a database. So let's say I want to know what the function is of this novel sequence. The classical approach is to build what's called a multiple sequence alignment. So given an input sequence that we want to understand better, we can use a search tool like BLAST or Hammer to search a database and find a set of sequences that are evolutionarily related across potentially disparate species. We can then align these sequences with the sequence alignment method, and this is shown on the right. So this is the simplest way to do transfer learning. So given our query sequence returned a bunch of hits to proteins that are all loop structures, we can be relatively confident that our query sequence is also going to exhibit this structure. So how do we capture the information encoded in a set of related sequences? For a set of aligned sequences in a single family, we can extract what's called conservation information. A position is conserved if it rarely changes across evolutionary homologs. And we can model this with a position-specific scoring matrix, or PSSM, which models each amino acid in the protein as an independent draw from a per position distribution. We fit this model by matching the first order statistics in the alignment. And we can represent the PSSM as an L by A matrix. So if you're afraid of equations, we have diagrams. Where this is an L by A matrix, where L is the length of the protein and A is the number of possible amino acids at each position. So here L is 4, A is 3. And in this alignment, we're very confident that position 2 should be red, because all of the examples we saw were red. But we're less confident about the distribution of other positions. So first order models like the PSSM and the independent sites model don't capture pairwise interactions. And if we want to capture coevolution, which is how multiple positions evolve together, we need to use a second order model, such as the Potts model. If you've seen the Ising model, this is a generalization of that model from statistical physics. We fit this model by matching first order occurrence and second order co-occurrence statistics in the family. And we can visualize the parameters of the Potts model as a symmetric matrix with L blocks of A by A, representing all pairwise interactions within this protein of length, in this case 4. So since there are 3 possible amino acids, we're going to have 3 by 3 blocks representing the interaction of a blue and yellow, a red and yellow, etc. And here, blue and yellow is the strongest interaction that's picked up by this Potts model, which is highlighted in green. So what about the rest of the sequence? We've only been modeling sequences from a single family with the models that we've shown so far. And what about higher order interactions, 3-way, 4-way, etc.? So to model multiple families at once and capture higher order interactions, we need a more flexible model class. Since proteins can be encoded as strings of characters, where each character is an amino acid, we can try to leverage the same techniques that worked in natural language processing to learn from documents to learn from protein sequences. This approach is part of a framework called semi-supervised learning, which combines labeled and unlabeled data. So here's an intuition for it. Let's say I'm trying to learn a classifier between blue and red. Here I've observed 2 labeled examples from different classes, and I want to draw a decision boundary between them. The one I've drawn with the dotted black line is a very natural choice given no other information. And I know it defines a classifier because I know immediately if I see a new dot and it's on the left, I'm gonna call it red. If it's on the right, I'll call it blue. But let's say I'm able to observe some unlabeled data. I realize that my data actually falls on 2 manifolds, and my decision boundary might need to look different. This is the goal of protein representation learning. By embedding all proteins in a high-dimensional space, certain manifolds or clusters become clear. And the representation here might lend itself well to, for example, classifying CRISPR-associated enzymes like Cas9, which all cluster together in this red cluster. So why is this a reasonable approach for proteins? The amount of raw protein sequence data has exploded as the cost of genetic sequencing has plummeted. And many of these sequences are coming from environmental DNA, sampled from extreme environments like hydrothermal vents and soil in Siberian tundra, etc. And the genes of the organisms that live in these environments are extracted, and the encoded proteins are deposited in a database like UniRef or UniPark. So these raw unannotated genomes are our unlabeled data. And we have many more orders of magnitude unlabeled sequences than we do labeled sequences. So for example, 500 million sequences in UniPark currently versus 200,000 in the PDB database of crystal structures. I feel like if you saw an inconvenient truth, this is the moment where Al Gore goes up on his little like scissor lift. So lots of unannotated sequences is analogous to the incredible amount of raw text available on the web. And some of you may have seen headlines about NLP models learning to answer questions and generate human text. The two basic ideas in models like GPT-3 and BERT are pre-training, which is the P, and the transformer, which is the T. And we'll come back to the transformer architecture, but we'll talk about pre-training first. So by training on 300 billion tokens, basically word pieces, scraped from all across the web, these models are able to recapitulate linguistic patterns like syntax and semantics. And GPT-3 achieved state-of-the-art performance on few-shot learning tasks where labeled data is scarce, like translations into a target language or sentence parsing. And since proteins can be encoded as sequences of characters, the language models can be ported over without too much modification. So let's look at how we do pre-training for proteins. The key aspect of a protein language model and of all language models is how it's trained. So how do you learn from this unlabeled data? We have to take our unlabeled data and turn it into some sort of prediction task. So here we're training using mass language modeling. Basically, we play fill in the blank, which as we all know is every robot's favorite game. And for millions of protein sequences, we draw a sequence, mask out a set of positions, and ask our model to predict what the mass position is going to be or should be. At each position, our model outputs a probability distribution over what it thinks should go in that position. And here it thinks lysine is highly probable. And we know what the correct answer should be. So we can then penalize our model with a cross entropy loss. And since this is a neural network and is fully differentiable, we can then update the parameters of our model. This is just a way of learning a probability distribution over a set of sequences and really doesn't rely on anything from linguistics. It's not like babies learn English from filling in Mad Libs anyway, so it's not unreasonable to think that this approach should work for proteins. So at each position, prior to outputting a distribution over amino acids, our model produces a per position representation of some dimension, D, which we can then use for other tasks. And during pre-training, we use this representation in purple to do mass language modeling. But we can take the same representation and transfer it to some specific task where we have a smaller amount of labeled data. And we can replace the downstream model from doing mass language modeling to now doing something task specific, like in this case, predicting the family of the protein that I've put in. Here it predicts a globin. This little sequence is taken from hemoglobin. So remember that if our representation is good, then we should expect it to improve the performance of a downstream classifier compared to a classifier taking in the input sequence directly. So we've broadly outlined two approaches to leveraging evolutionary information, the retrieval approach and the storage approach. So in the retrieval approach, we took a query sequence, scanned it against a database, constructed a multiple sequence alignment full of homologs, and then extracted conservation and coevolution information. In contrast, in the storage approach, a pre-trained model will store evolutionary information directly in its parameters, and we don't need to query a database. You can just query the model directly. So let's summarize this section. Reuse is pervasive in biology. There are hundreds of millions of unlabeled sequences. Single family models like PSSMs, POTS models capture important evolutionary information from multiple sequence alignments. And protein language models trained on large protein sequence databases store evolutionary information in their parameters. Armed with these concepts, we're going to enter chapter 2. So here I'm going to argue that protein language models have come of age and do so by focusing on the task of structure prediction. A number of different groups in 2019 arrived at the conclusion simultaneously that language models could be applied to protein sequences. But the state of the field was such that there were many different protein representations that were difficult to compare. They trained on different pre-training corpuses, had different compute budgets, and different downstream tasks to evaluate their performance. And we want to be able to compare these because they have all of these choices might be important. Things like the architecture of your model or using auxiliary information from structure or multiple sequence alignment might be important for deriving a good representation. So what was the best approach? And in order to figure this out, figure out which representation was better, we had to fix the corpus, fix the compute budget, and fix the task, and then modify other design decisions. So this is work that I did with colleagues Nick Bhattacharya and Roshan Rao, which we called TAPE, Task Assessing Protein Embeddings, where we attempted to benchmark these protein language models against one another. We fix the corpus to be 31 million protein domains in PFAM, fix the training time, and most importantly, evaluate it on a common set of 5 tasks. And each of these tasks was chosen to be biologically relevant and span a number of interesting modeling objectives, from structure prediction, homology detection, and protein engineering. Using our language from earlier, these tasks include those related to discovery, classifying novel sequences, and engineering, optimizing. So a lot of focus will be on the task of contact prediction. So I'm going to start by talking about that task in particular. Contact prediction is a binary prediction task where the goal is to figure out which pairs of residues are within 8 angstroms of one another in the 3-dimensional structure. And contacts can also be represented as a contact map, which is an L by L matrix, where a 1 in the matrix position ij corresponds to positions I and j being within 8 angstroms of each other in the 3D structure. So here, the green dots in the structure correspond to the green dots in the matrix. And if we can predict a contact map for a novel protein, we can make a rough estimate of its fold and therefore gain some understanding of its function. So in TAPE, we attached a downstream classifier to our language model, trained on a set of structures, and looked to see how our pre-trained representation improved contact prediction. We trained 5 different types of pre-trained models, spanning different architecture choices. And each of these representations we evaluated on all of our tasks. So what did we learn? Without any pre-training, taking in the raw sequence results in a poor contact map. Here, the true contact map is shown on the left, which is some bacterioferritin co-migratory protein, and the predictions are shown in the center. So true predictions are going to be in blue, whereas false predictions are going to be in red. And darker colors represent more certainty from the model. And we did find that pre-training helped. In this example, the pre-trained features helped us pick up some additional long-range contacts, like the ones circled in green. But when the results really rolled in, we were a bit dismayed. Because while pre-training wasn't entirely unhelpful, simple bioinformatic features like a PSSM, which remember is just the frequencies of occurrence of amino acids in that multiple sequence alignment, ended up being much better features for prediction of contacts. The contacts are sharper, have fewer false positives, and really this is kind of a recurring theme in this line of work, which is beating multiple sequence alignments is hard. And Nick even made this meme at the time saying that we had entered the field at just the wrong moment. Too late to explore eukaryotes, too soon to explore the rest of the proteome, but just in time to lose to classic bioinformatic features. And despite this devastating meme, you'll be happy to know research progressed forwards. There were 2 major breakthroughs, which were the realizations that 1, protein contacts can be extracted directly from protein language models, and 2, contact extraction can be used as a benchmark on its own. So to understand how we do this, we have to understand something about attention, which is the key operation inside of protein language models. So we're going to get to the T, the transformer. There are a few different neural network architectures that can be used for protein language modeling, but most are based off of the transformer due to its success in natural language. And the key operation that makes these models tick is what's called self-attention, which I'll just call attention for simplicity. In an attention layer, each input is converted into a key vector, and each output position is associated with a query vector. And to determine the attention map for the output at position 4, we compute this query vector, keys for each input position, and take a dot product of the query with each of the keys. The value of this dot product gives us the edge weights, with here darker being stronger or larger. And we can represent the attention at position 4 by this vector of length 7, representing the edge weights to each of the input positions. And because each output position is associated with a different query and a different attention map, we can then visualize the full attention map for this layer as an L by L matrix. And this is going to be the source of our contact maps. In the full model, we have multiple heads of attention, and each head can determine how to allocate its edge weights. So a head could specialize, for example, in looking at interactions between positive and negative residues and learn to pick them out. This model then repeats the procedure for multiple layers, refining the representation at each layer. So self-attention followed by what's called a feed-forward layer, repeated as many times as your biggest data center will allow. So in a paper by a team at Salesforce, they found that certain attention heads did specialize, and they specialized to picking out protein contacts. And we can see that in the early layers of this transformer, they're mostly uncorrelated with contacts. But as we get to the final layers, there are some heads here that exhibit really high correlation with protein contacts. And to extract contacts from the transformer, we follow this procedure. Find this set of heads, which you do by taking a held out set of structures and sequences, and trying to find the heads that are most correlated. And then to do contact prediction on a new query sequence, we input it into the model, pick out the attention maps from those heads that we've selected, average those attention maps, and then take the top L highest magnitude positions in that attention map to get our predicted contacts. Extracting contacts from evolutionary models is how Potts models initially actually gained a lot of traction, going back to work in the early 2010s. So to extract contact from Potts model, we again look for the strongest couplings in our matrix. So here we find the blocks with the largest sum of squared elements. In this case, the block corresponding to the interaction between position 1 and 4 is the highest, so it will be indeed predicted as a contact in our extraction procedure. So let's talk about how we evaluated contact prediction in tape. So we evaluated all of our representations in the semi-supervised setting. So this is from the results from before, where we pre-train a language model and use that pre-trained representation as input to a downstream contact map predictor, which is trained on some existing structures, and it learns to do this conversion. But the understanding of how to extract contacts directly from the model allowed us to coalesce around another evaluation, unsupervised contact extraction, which has a few key benefits. In this problem, the model never sees any structures at all. And this is really nice for its simplicity, because the performance of the language model doesn't depend on the capacity of the downstream predictor. They're also really good data sets. These are structures with associated sequence alignments. And this is important because data sets for protein engineering, for example, really vary in quality. And contact prediction has very interpretable metrics. How many of your predicted contacts were really in the structure? You got 7 out of 10 right, you get precision at L of 70. And we have a very strong baseline. So this is also very important, that we have a baseline that we want to beat and that we know does really well. So if we can beat Potts models, we know we've learned something important. So with this new benchmarking task in hand, we could see clearly that some models were vastly improved. And here I'm showing the performance improvement from the transformer we trained in tape compared to ESM1B, ESM being short for evolutionary scale modeling, which is a language model from Facebook's research team. Each dot here corresponds to a different predicted structure. And the more dots that are above this dotted line, sorry, on the x-axis, we have Potts model, on the y-axis, we have our transformers. And each dot that falls above the dotted line means the transformer is outperforming the co-evolutionary model. The more dots that are above this dotted line, the more families on which the language model outperform the co-evolutionary model. So the tape transformer is pretty much completely outmatched, except for on structures with the very smallest MSAs, whereas ESM1B is competitive with Potts across a wide variety of protein families. Remember, the tape transformers struggled to even outperform PSSMs, and now protein language models are looking competitive with even stronger co-evolutionary methods, retrieval methods. So work that I did with Nick and Sergey also confirmed that protein language models consistently outperform single family models on small MSAs. In general, contact extraction performance is going to be correlated with the size of your MSA. The more sequences you get to observe, the better you do at extracting coevolution. The red line here is the Propert BFD transformer from another group at the Technical University in Munich. And on small MSAs, the Propert BFD model significantly outperforms these single family models, which indicates that there's some additional signal that this model is able to leverage compared to a single family model. It's able to use information from other families, from other proteins, potentially unrelated, and use those to resolve the contact maps further. So what changed? How did these models come of age? Sometimes it hurts to say it, but bigger training set and bigger models. It's a bitter lesson in AI research that's been repeated across a wide variety of fields. But yeah, that was a major difference between tape and Propert BFD. So in tape, we trained our language model on 30 million domains from PFAM, whereas Propert BFD was trained on 2.1 billion sequences from the big fantastic database. That's the BFD, if you're wondering what the the models also increased in scale by a factor of 10. So since then, models have also continued to increase in scale. One subtler change is that folks threw out this idea of training on individual domains, which are portions of the protein that are ordered, and instead trained on full sequences, which we thought that limiting the amount of disorder in the training set would be helpful for language models, but it doesn't turn out to be the case. More data seems to be better. So I'm going to close this chapter on structure prediction with a more recent research development. We've seen that protein language models can store structural information implicit in sequence databases. So let's go back to the supervised structure prediction problem. This is what you all came to see, right? But the biggest story in supervised protein structure prediction is AlphaFold. So at CASP14, AlphaFold achieved levels of structure prediction performance not expected for another decade of research. It was named Nature Method's Method of the Year, won DeepMind the Breakthrough Prize, and caused academics working on protein structure prediction to experience strong empty nest syndrome. I love this quote. It feels like my child has left home. It crushed every other method. Without doing a deep dive on AlphaFold, I want to highlight the part where protein language models come in. So let's go back to the first version of AlphaFold, which dominated the previous version of CASP, CASP13 in 2018. To predict the structure for an input sequence, first they use a database query to construct a multiple sequence alignment, extract coevolutionary features using a POTS model, and feed those features into the rest of the model to predict structures. In AlphaFold 2, the MSA gets mapped or passed directly as an input to the model and is processed by this evoformer or evolutionary transformer before being passed downstream to the structure module. So in addition to structure-based losses, they included a mass language modeling loss by masking positions out in the input MSA. And new results from this year actually at groups at Facebook rebranded to Meta. So Meta and Harvard show that the MSA retrieval step can be replaced with a language model. So that now these features combined with the downstream structure module achieve competitive structure prediction performance results to the original retrieval-based AlphaFold methods. And this one is hot off the press as of yesterday, Roshan texted me. So just to give you a sense of what it's like to work in this field. Because language models encode sequences with a single forward pass, it's much faster than retrieval-based methods to featureize input proteins. And this allowed Meta AI to generate 600 million structures from metagenomic sequences and release them yesterday morning in the big database, along with an API where you can just put in a sequence and get out a structure in seconds. So let's summarize chapter 2. Protein language models have come of age in a major way. They're competitive with coevolutionary models on unsupervised contact extraction and beat single family models in this low data regime, which is important because if I want to make predictions for a protein where there's no good alignment, like a viral protein or this random jellyfish protein, this is where we really care about alignment-free methods. They can also be used as features to improve downstream structure prediction. So what's next? Let's go back to where we started, protein design. Structure prediction is an amazing sandbox for model development and is an important problem in its own right. But the next frontier is how to take these models and turn them back on the task of engineering novel proteins, ones that nature could never dream of. So recall that we had two goals for ML-guided design. The first was to discover new solutions to biochemical problems, and the other was to optimize them. There's been a ton of work on these topics, and I can't claim to comprehensively cover it, but I'll highlight some motivating results that use protein language models, starting with optimization. So the problem of variant effect prediction looks like this. For a given sequence, I can look at all the possible single substitution mutations that I can make. So for this particular region of the GFP protein highlighted in pink, I can mutate the existing amino acid to one of 19 possible variants. And for each of those variants, I can score their functional effect. So in blue are variants that maintain the fluorescence activity of GFP, whereas red, those mutations destroy the activity. They're called deleterious. And there are several data sets that have these experimentally verified functional effects that we can use to evaluate the effectiveness of a predictive model. And given a trained language model, I can actually do variant effect prediction. So how do I do this? For a given input sequence, I mask out the position that I want to mutate and look at how probable the model thinks each amino acid substitution is at that position. And the log ratio of these probabilities relative to the wild type here, which is L, gives you a score which you can use to rank the mutations. Here, V is given a relatively high probability by the language model, and so it's predicted to be a functional mutation, and A is given a relatively low probability, so it's predicted to be deleterious. And across a wide range of experimental data sets of variant effects, ESM1V here in blue, again from the Meta team, showed that it was competitive with single family models, which are shown in gray. And this means that given a starting point for protein engineering, we can use our language model like this one to avoid introducing deleterious mutations. So let's turn to discovery. The goal of discovery is to find something novel, something that nature maybe wouldn't have come up with itself, more than a few mutations away from known solutions. Progen, so this is another model. Progen is a model from Salesforce that leverages another NLP technique, namely that of control tags. So given documents with genre labels like politics, sports, or horror, a language model can learn to do next token prediction and generate sentences that make sense in that genre. So here, it asks to generate something from sports, says the Red Sox defeated the Yankees, proving that this language model is from Boston. The analogy in proteins is to use the protein family as a tag to control the generation. And instead of doing next token, it's going to do next amino acid generation and output sequences that are consistent with this protein family label, like immunoglobulin or lysozyme. So collaborating with researchers at UCSF, the Salesforce team crystallized the first AI-generated protein, a lysozyme, which is a very well-studied class of antibacterial enzyme. And here it is in the PDB, lysozyme 56 from deep neural language modeling. There are still a lot of outstanding questions as to how best to use these language models for protein design. And even with the successes that I've highlighted, which I hope are the first of many, this problem is by no means solved. And it's not to say that language models are the only models that can perform these tasks. So retrieval-based methods can also do all of the tasks that I just described. But I believe that this arms race between storage and retrieval methods promises to be really fruitful for the community, pushing progress forward in structure prediction, variant effect prediction, and sequence generation. The applications I'm most excited about right now are about taking these methods into the lab and verifying their predictions. And already some biotech companies have put protein language models into their core technology platform. Absci and AbloBio, just to name two, are both aiming to leverage them for antibody design. So these models have started to wander beyond the halls of academia, and I decided to follow them. So I'm really excited to be joining the team at Google X, working on a moonshot in protein design. So I've been really blessed to be able to think about these problems with a lot of great collaborators here at Berkeley, at Google, at Harvard, elsewhere. And so we've come to the end of the talk, and I'd like to take a moment and thank the people that helped make these last 5.5 years possible, get me through the hard times, and we're there to celebrate the good times. So first, I want to thank my committee, Ian and Nyla, for being so supportive, and especially Ian, who isn't here, I don't think, but he was willing to dance to Rihanna in my Protein ML reading group, and for that, I'm grateful. I have to thank Yoon, of course, who from the looks of his Zoom background has spent a little time in the library of Babel himself. Thanks for modeling the consummate scientists, for trusting me and always bringing that curiosity, the little boy in the candy shop. That's what drew me in initially, and that's what keeps me going. Like Yoon said, he was there when I first learned about entropy as an academic baby, and has seen me grow into a graduate student who still has really way too many questions about entropy. I have to thank Nick. Nick and I have worked so closely together that some people struggle to tell us apart. We spent countless hours working together in the lab, stealing Yosef's lab Thai food, and had multiple week stretches where we didn't turn our Zoom cameras off. Nick has a great sense of humor. He's incredibly patient, and he'll also wake you up in the morning saying he rewrote the whole paper overnight. So for Nick, any concept is just one afternoon with a textbook away from being understood, and it's such a refreshing approach to research and to life. I was really lucky to meet Roshan when I did. This is a man who knows how to enjoy the good things in life: red wine, homemade paneer. He brings the calm confidence of a man who knows he's about to execute. His code is as aesthetic as his homemade meals, which are Michelin star. And I continue to learn a lot from working with him, and I forgive him for every time he called me Nick. This is my favorite picture of Roshan with our tape poster in rainy Vancouver, finally where it belongs, in the garbage can. When the pandemic hit, a lot of things became further away, but one of the bright spots was our completely grassroots Protein BERT research group. So it was Sergey at Harvard, Eustace at University of Washington, Peter at Cold Spring Harbor, and this was a really productive and amazing group. And Sergey is this rare form of wizard where if you ask him a half-baked question, he already has 100 slides on it and 2 alternative theories of his own. So I can't wait to see what university you end up at. Спасибо большое. I spent most of my 4th year optimizing an enzyme as an intern at Google. And I was blessed with some amazing mentors: Lucy, David, Atish, and Abby, some of whom are here. These are folks who are really excited about the work that they do and always willing to lift each other up. So I'm really glad to be joining their team. Shout out everyone in the Song Lab, past, present, and future. Our lab is an inspiring and sometimes insane place to work, where everyone works on something completely different. But everyone is incredibly sharp and also incredibly humble, and I know that that spirit will continue. I have to thank the Pomodoro Bros, or AKA the work from homies, Nick, Elizabeth, Sanjeet. These folks would log on with me every morning for weeks in the era of remote work and help keep me on task through 25-minute Pomodoro work sessions. And if you don't have a work from homie, you should get yourself one, especially one who'll show you the zucchini they just harvested. So last, I'll thank my family, some of whom are here. My mom, my dad, my uncles, which I call the ambassadors to my left and right brains. Thanks for always cheering me on. And I hope you understood something today. And if you didn't, ask Uncle Mark. So with that, I'll stop, say thank you, and take any questions.